welcome to the Journal of Zoology podcast. I'm Elina Rantanen. This episode features articles published in the journal from June to August 2012. We will hear about analyzing the structure of a bird song, learn whether the blue throat patch of European green lizard males is an honest signal of individual quality, and explore resource and habitat use of spider monkeys in Costa Rica and grazing ungulates in African savannah. Birdsong is often used for species recognition, but analyzing the structure and variability of a birdsong could unveil a lot about the species' evolutionary and biogeographical history. Lauren Benedict at the University of Northern Colorado, together with Rory Bowie from the University of California, used song recordings of the rattling cisticola to analyze song features in the species, and I spoke to Lauren to find out more about their research. First of all, thank you very much, Lauren, for talking to us about your intriguing study. So, why is it useful to analyze the song structure and variation of a bird such as the rattling cisticola?
Well, I think it's interesting to analyze these song structures for a couple of reasons. I think generally it's really interesting to look at bird song structures, first because it's a really useful way to identify species, and then it also gives us a sense of the diversity of phenotypic traits, in this case song, that a single species can express both at the individual level and across the geographic range of a species. And then I think it's also really interesting because what we see when we look at these song structures is kind of evolutionary outcomes of processes that create patterns that create diversity in phenotypic traits. And in particular in cisticolas, I think this is really interesting because its song in this group has been selected for to be very diverse, and morphology has really not. So when we look at cisticolas, there are 40 to 50 species. It's unclear exactly how many species because we think that there are still probably cryptic species out there. And there are 40 to 50 species that not all, but almost all of them live in Africa. And you can have, say, one species that overlaps with maybe 30 congeners, and it looks morphologically almost identical to all of them, but it sounds very different. So song is really a great trait among cisticolas for trying to understand what they use to indicate species affiliation and how they maintain reproductive barriers between species.
Yes, birdsong analysis sounds like a very useful tool for a multitude of research questions. How did you then go about analyzing the song structure and variation in the species?
Well, what we wanted to look at, our main goals with the paper were to try to understand the form of the song and how that song was used by one particular bird, individuals, and then how that song might vary across the range. And so kind of our main questions that we were trying to get at with this analysis was to understand if it did indicate species affiliation. And so we were predicting that there was likely, because this species, the rattling cisticola, overlaps with so many of its congeners, there was likely some kind of selection, stabilizing selection, for parts of the song that might indicate affiliation and species identity. At the same time, this song has been reported in literature to be one of the more variable cisticola songs. And so we thought that there was likely some kind of selection for variability of other parts of the song, and that we also had good evidence that might relate to sexual selection and territory defense because they live in groups and they use song to defend territories of different qualities. And so songs are used in variable ways when they're defending territories. So there's likely some kind of selection for variability as well as this selection for stability. And so what we did then was we got sound files from sound libraries. And our main sources were the British Museum, the Macaulay Library of Natural Sounds, and the Ditsong Museum. And we obtained about 61 files from almost the entire range of the species. And I have to say here, it's really nice to use, I think, library sound archives because you can get such a broad sampling that if we wanted to get, it would be very difficult and time intensive to 61 different sites across sub-Saharan Africa. that together cover something like 4,000 kilometers.
Uh-huh.
And so we got all of these sound files, and then what we did was we looked at different types of the song, or different parts of the song, and we found that they have a song that has kind of a typical structure, where they have these introductory notes and then end phrases. And so we characterized the form of the introductory notes, we characterized the form of the end phrases, and then we looked at the way that they pattern songs and how they put those introductory notes and end phrases together. And then we also looked more specifically at some of the frequency and time parameters that describe those different parts of the song within individuals and also across the geographic range.
Okay. And what did you discover as the characteristic features of the song of Rattling Sisticulus?
Well, all of the songs really did adhere to a really typical pattern that people had described before, where they use an introductory note that they'll repeat 1 to 3, occasionally 4 times, and then they follow that by an end phrase. And usually those introductory notes are a little more simple, and the end phrases can be much more complex, that can include trills and lots of different shapes. And we found that they do deliver them in that typical pattern, and there was a lot less diversity in the introductory notes. There were 3 introductory note types that we found. There was one that's kind of a frequency modulated sweep, one that is a broadband buzzing sound, and one that is a harmonic stack. And those 3 introductory note types might be given all by a single individual. And at the same time, we looked at these end phrases, and there were 77 different end phrases that we identified, and very few of them were given by multiple individuals. And that's probably related to sampling. There were some that were given by multiple individuals. But when we were looking at the songs of a single bird, what we found was that they would continue to give new end phrases right up to the end of the tracks that we had. And so we think that these end phrases then are very variable, and there are probably many more than 77 different types across the population. We just didn't have enough sampling to get at that. And there might be potentially almost infinite variety in the end phrase types within Rattling Sisticulus.
Aha. And did song features vary by geographic location?
Again, we looked at the first and second parts of the song, and among those introductory notes, I said there were 3 different types, and 2 of those types were given across the entire range of the species. And one of them, though, was unique to southwestern Africa. So we only had 5 sample points, I think, from the kind of western side of sub-Saharan Africa. And in all of those, they gave this harmonic stack introductory note. And so there's clearly a break there between these groups of birds that use that particular stack introductory note and the rest that don't. And I think it would be really interesting to follow up to try and locate exactly where that divide is, where that break happens, that they start using the stack as you move west across Africa. And that would require maybe doing some interesting playback experiments too. I think you could test to see if, for example, eastern birds recognize that harmonic stack note as the note of a rattling sesticula. So there's a clear geographic difference there. Otherwise, though, the introductory notes were really very stable. So the other 2 types of introductory notes, those sweeps and the buzz notes, really didn't change at all across the entire 4,500 kilometer geographic range that we were looking at. They were very characteristic and very similar in the way that they were used, and also very similar in their time and frequency parameters across this very large range. And then the second half of the song, though, we saw a very different pattern. In the end phrases, there was really, as I said, a lot of variability in these end phrase types. And in addition to having variability in types, there didn't seem to be any clustering of end phrase types. So we saw geographic variation not in a way that you might expect if there were local dialects, say. There were not certain areas that all sang the same end phrase type. In fact, when we saw different birds singing the same end phrase types, they were often very widely distributed in space.
Okay.
Yeah. So I think it's a really kind of surprising there. It's not sort of a dialect system. There's a lot of variability, and it didn't seem like neighbors were sharing song types, although we didn't have extensive sampling really to test that very well. But one of the other things we did find when we looked across geographically and elevationally, we found if you go across the geographic range of the species, as you move kind of northeast towards the equator, you find that the birds are singing end phrases that have higher high frequencies and wider frequency ranges.
Ah.
So there seems to be some kind of clinal change as you move across that geographic range. There is not a corresponding elevational clinal change, just that geographic change. So there are sort of large-scale geographic patterns that we thought were interesting.
Yes, it is fascinating how you found such variable geographic patterns in the different components of the song of this species, and this playback experiment you mentioned would indeed be very interesting. How would you then suggest that the found song features of this species may have been shaped by ecological factors or evolutionary processes?
Yeah, that's one of the main things we were really interested in thinking about, how this pattern does represent those evolutionary forces. And that clinal pattern that I mentioned, that change across the geographic range in the end phrase high frequencies, is one that we found really interesting but sort of puzzling. We don't have a great answer for that, but that certainly could be a result of ecological gradients across the African continent. It also could be a result of the fact that as you move away from the equator and temperatures cool, animals often tend to have larger body sizes at cooler latitudes, and so they might be singing lower frequencies at those higher latitudes, I should say, away from the equator, because they have larger body sizes. That's a possibility that we would have to look into, though, and the lack of an elevational gradient doesn't really support that. So that's a very good question. But the things that we do feel like we had sort of good conclusions about were our original predictions that stabilizing selection should promote some elements of the song that stay really constant across the geographic range and help to identify species or help to identify individuals to a particular species. And so we found that in the introductory notes, those introductory sweep and buzz notes that were so constant across the geographic range might be really good indicators of species affiliation. And it's kind of interesting or nice that it occurs right at the start of the song because it's sort of an alerting information that indicates to the listeners what species has begun to sing. And then the following information, the end phrases, might convey more fine-scale information about that individual's quality or that individual's identity. And those end phrases then were, of course, much more variable, and so might be better able to vary with quality and individual differences.
Sure, that makes a lot of sense. And how could this kind of song analysis help the identification of other problematic bird species?
Yeah, well, I think that if we stick with our discussion of sort of the genus, the sticklas, as I've said, are such a large group that are so difficult in many ways to tell apart. Understanding the song pattern here, I think, is good because this is one of the species that is most widely distributed and also is kind of often discussed as having a very variable song. But if you look closely at it and you start to understand, yes, it's variable, but it's only certain elements that vary, and you can focus on the fixed elements, then you can find commonalities across the geographic range. And so we can look at this in all kinds of species, and we have done this in at least one other species of Cisticola. In a study of a congener, the red-faced Cisticola, my co-authors and I, we looked to see how their song patterning might be either stable or variable. And they also had a really fixed pattern of song delivery. It was different from the rattling Cisticola's pattern of song delivery, but it was a very good indicator of species affiliation. And so starting to look at these commonalities between them, I think it brings up an interesting question of whether song pattern delivery is a feature of bird songs that is really important and tends to be more often stable and an indicator of species identity, whereas the actual form and frequency modulation of the notes might be more often variable, and there's kind of a little more wiggle room or room there for evolution and sexual selection to play with changing those features, but keeping the pattern stable as an indication of species affiliation. And I think there's kind of a cool analogy with human language there, where if I for example, don't understand French or Japanese, which really I don't. But if I hear someone speaking either of those languages, they're delivered in a way that I can recognize what language that person is speaking because of the patterning of delivery, even though I don't understand the exact frequency modulation or the form of the words. And
sure.
You might have an interesting analogy there with birdsong.
What a great analogy indeed. Well, thank you very much, Lauren, for this interview, and I look forward to seeing your future research. You can read the full paper, entitled Rattling Testicular Song Features and Variability Across Sub-Saharan Africa, in the June issue of Journal Zoology. Sexual selection has fascinated and puzzled zoologists since the days of Charles Darwin, and nuptial traits signaling individual quality have a prominent role in this evolutionary game of attracting or choosing mates. European green lizard males develop a blue nuptial throat patch, and Orsolia Molnar and colleagues at the Laurent University examined the relationship between the color of this throat patch and the quality of the male in terms of fitness-related physical traits. I spoke to Orsolia to find out more about their study. To start with, thank you so much for telling us about your research on this fascinating subject. So, why did you choose to investigate the relationship between male quality and the color of their nuptial throat patch in this lizard?
Well, we originally started to investigate this because we only knew that this lizard species elaborated intense blue throat patch color. And our first question was whether this coloration was actually nuptial. And so we experimentally tested whether it affects female mate choice or contest competition. And we actually demonstrated that they are both influenced by this nuptial throat patch. And so our second question was whether this was an honest signal, that is, the information content was really the question, and whether it was a multiple signal. So these were the primary issues we were trying to test. And in addition, lizards are very suitable model species to do such research because they're ectotherms. And ectotherms have lots of temperature-dependent physiological processes. And since the daily time available for maintaining appropriate body temperature is limited, then this way the time spent with acquiring high temperature actually affects individual traits. And given the larger variance in duration of this appropriate body temperature maintained, the larger variance is actually shown in individual traits as well.
Aha! So this lizard sounds like an ideal species for studying a nuptial trait. And how does your study fit into the wider theory of sexual selection and its prominent hypothesis?
Well, sexual selection actually has been investigated in a wide range of species. And interestingly, lizard and reptile species have only recently begun to come into these studies, although they show a very wide variety of sexual dimorphism and dichromatism, and males compete for the female preference. And this way, mate choice can be based on lots of types of signals, including visual, audio, and chemical signals. And visual signals have been very rarely demonstrated in lizards. And since we have demonstrated UV color-based signals in these lizards, we were very interested in the information content of this signal. Now, the two hypotheses that possibly can work here is either the Zahavi's handicap hypothesis, which says that this coloration should be an honest signal, and the other is the Fisher-type sexy sun hypothesis, saying this is only an attractive ornament, and it has no actual information about the owner's quality. Females are only preferring this because it's attractive and conspicuous. And through our studies, we demonstrated that this signal is costly and indeed signals better survival and health. So we think that we have demonstrated and have gained evidence for the handicap theory, saying that this coloration actually fits into the Zahavi's hypothesis, and females can actually acquire reliable information by estimating this signal.
Okay, so to investigate the honesty of this nuptial signal, what traits did you measure in the male lizards and why?
Well, obviously, we measured throat color since that was the main question. We wanted to answer what throat color actually means. Throat color was measured with a spectrometer, and there were 3 variables recorded. The first one was total brightness, which is the average reflectance between 320 and 700 nanometers. And the second 2 were the UV and blue chroma, which are the proportional average reflectance in the UV and the blue range, respectively. We also measured body size. Full body size was characterized with SVL, that's the snout to vent length. This is a pretty commonly used index in reptiles. And head size, which was characterized by head length, head height, and head width. And considering reptiles, lots of reptile species, including lizards, show infinite growth throughout their life, a larger size can represent an older age, saying that larger animals can actually show a better survival. Finally, we measured body weight, which was measured by an analytical balance. We measured the number of ticks, and we measured the number of femoral pores on the right and left side, because the difference between the number of these pores has already been demonstrated to signal femoral fluctuating asymmetry. And interestingly, in this species, we found that this actually represents directional asymmetry, which might be a sign of handedness, but obviously warrants further investigation.
You mentioned that you counted the number of ticks on the males. Could you elaborate what was the purpose of this measure?
Well, the number of ticks can actually signal, obviously, parasite load, and through this, the resistance to infection, or the lower number of ticks might actually mean a better ability to avoid infection in some way. Obviously, a general health status.
Yes.
So you presume that the individuals with the lower number of parasites are in a better health condition compared to those with a higher number of parasites, a larger parasite load.
Sure, that makes sense. Did you then find significant relationships between these physical traits and the nuptial patch color?
Yes, we found numerous connections between the components of the throat color and individual traits. We found that individuals with a higher UV reflectance, that is higher proportional UV color, had more femoral pores on the left and were in a worse condition than those with a lower UV reflectance. And bright patch color actually was associated with larger body and head size and less ectoparasites.
And what do these results reveal about the function of this nuptial throat patch in intron intersexual selection in the species?
Well, considering that male nuptial coloration was shown to carry information about several individual fitness traits, we have to assume that all of these are possibly taken into account in both inter and intersexual selection. UV has experimentally been proved to affect female choice and male contest, but nevertheless brightness is also very likely to be evaluated in both of these processes. If we look at the negative connection between UV color and condition, UV is possibly presumed to be costly, and therefore only the better quality males can afford the costs that are raised by this high coloration, high intensity coloration. And because these costs are raised by the conspicuousness of this color. So if you look at the owner being more conspicuous to predators and other males, they have to bear a higher predation risk and more male interactions or contests, and therefore only better quality individuals can afford to elaborate this high intensity signal. Positive connection between UV and asymmetry can refer to, as I said before, handedness. This femoral pore asymmetry might signal a systematic developmental bias towards one of the sides, which might have further advantages. But this is the part where we have to do further investigations for me to be able to say anything more precise. Brighter color associated with larger size and less parasite load. These are both signaling better quality, better survival, better health status, and referring to the handicap hypothesis, better quality is actually worth signaling in this species. Large size can not only mean the good genes that are given onwards to the offspring, but better survival, considering infinite growth in lizards, and lower parasite load can either signal higher resistance or this better ability to avoid infection.
Mm-hmm.
Taken together, nuptial throat patch color is not only an honest signal in European green lizards, but also acts as a multiple signaling system and gives detailed information about the owner for both potential reproductive partners and competitors.
Yes, so the females and male competitors are able to get honest information about the quality of the male based on its throat patch color. What would then be the future focuses of your research based on these results?
Well, having found that this signal is a multiple signaling system or part of one, we are now interested if the attributes of males' territories are also represented some way in this signal.
Mm-hmm.
And so we investigated the possible connections between spatial strategies conducted by individuals and the color variables from which the data we are currently analyzing. And further projects will aim to discover if females alter their investment into their offspring based on the sire quality, and whether the level of oxidative stress shows covariance with partner quality, and if it's inherited by the progeny. Both questions are being experimentally tested in 2 long-term research projects we are now organizing.
Aha! That sounds really interesting, and I look forward to seeing your results. You can read the full paper, entitled Individual Quality and Nuptial Throat Color in Male European Green Lizards, in the August issue of the journal. It is vital for effective species conservation to locate and protect the essential habitats and resources for the species. Norbert Asensio from Mahidol University with colleagues carried out a 4-year study on the space use of spider monkeys in Costa Rica to identify so-called core areas within their home range and to investigate habitat quality in these areas. To find out more, I spoke to Norbert about their research. To begin with, thank you very much for talking to us about your impressive study on these spider monkeys. So, what has been previously known or assumed about the significance of core areas in animals' home ranges?
Well, because core areas are the regions or the home range that an animal or a group of animals use the most, it is assumed that they should be of more biological importance or more ecological quality, if you will, compared to other parts of the home range. For example, they should have more places for shelter, like sleeping trees, or more important food resources, like fruit trees, or any other item that is critical for a given animal species. So it is also thought that these areas constitute the minimum area an animal needs to survive. So if something happens to the, let's say, non-core area, the area that is actually the rest of the home range, say a fire or any human activity damaging it, everything will be fine as far as the core area is untouched.
Aha! And why did you want to compare habitat quality between core and non-core areas in the home range of these spider monkeys?
Although core areas are assumed to have more habitat quality, as I was explaining before, and this is something that makes complete sense, it has been very difficult for us to find studies that actually did this important comparison. That's why we decided that it was necessary to do the test. And why in spider monkeys? Well, spider monkeys are very dependent on areas of primary forest, areas of good quality that are hard to find in a dry tropical forest where we did this study. Spider monkeys are highly frugivorous. They're very dependent on ripe fruit, and they occupy large home ranges compared to other arboreal primates of the same size, like gibbons or howler monkeys. So because of these reasons, we were expecting, first, clear, distinct core areas of spider monkeys living in a dry tropical forest. And second, we also were expecting to find more critical features for spider monkeys in core areas, such as sleeping trees, food trees, and patches of primary forest compared to the rest of the home range.
Yes, it is indeed important to verify this assumption about higher habitat quality in core areas with actual data. How did you then collect your data?
We follow a group of spider monkeys for about 10 complete days a month during 4 years. We did this in the Santa Rosa sector of the Guanacaste Conservation Area in Costa Rica. The geographical location of the spider monkeys was collected every half an hour with a GPS. We also collected the location of all important food sources and the sleeping trees they used. And finally, we collected information about the different habitat types, combining our knowledge about the forest and a high-resolution satellite image in order to create a land cover map showing the different habitat types.
Well, it must have been wonderful following these animals in the forest. So, what did your results reveal about the habitat quality of the core areas?
We found 5 different small core areas across the home range, and we also found that these core areas had higher densities of food trees, higher densities of sleeping trees, and also higher proportions of primary forest. However, these core areas by themselves only represented the 30% of the total food trees and the 60% of the total sleeping trees in the home range. And of course, not all the primary forest was included in the core area. Therefore, although these are pretty high values, 30% and 60%, they were not enough, on our opinion, to consider that core areas alone represented the minimum area this spider monkey group needed to survive.
Aha! And what significance should core and non-core areas then have in the conservation of spider monkey populations?
Well, core areas are important because they have high densities of critical resources. That's what we found. And therefore, they are key to understand spider monkeys' ecology and habitat use, and therefore they're important for conservation. However, we have to be very careful, thinking that core areas are the only areas that need protection, and so the rest of the home range is unimportant. After all, core areas didn't contain all the food resources, at least in this study, just an important part of them. The rest of the forest must be also important, not only because contains other sleeping trees and other food trees, but also because it has the necessary habitat to be able to move between core areas for spider monkeys. And moreover, there could be key features in the home range of the spider monkeys that don't need to be necessarily associated to core areas. For example, we know of some water holes that the spider monkeys used, but because they use these water locations for short periods of time and only a few times a year, they didn't lie within the core areas. And these water locations are certainly important for spider monkey survival. So what we recommend is that the conservation initiatives, at least concerning spider monkeys living in dry tropical forests, should focus not only in the core area, but also in larger tracts of the forest, like the home range itself.
Well, thank you very much, Norbert, for this interview. You certainly have provided valuable data and important recommendations for the conservation of spider monkeys. To find out more, you can read the full paper entitled "Spider Monkeys Use High-Quality Core Areas in a Tropical Dry Forest" in the August issue of Journal of Zoology. How can rare species coexist alongside abundant species with similar diet without being excluded through competition? Norman Owen Smith and colleagues at the University of Witwatersrand investigated habitat and resource use of the rare sable antelope and the abundant zebra and buffalo to uncover differences in resource use between these grazing ungulates that could explain how they can coexist in the African savannah. To begin with, thank you very much, Norman, for giving this interview on your interesting research. So, your paper is about coexistence of rare and abundant species of grazing ungulates. Could you tell us about the ecological concepts of species coexistence you used as the basis of this study?
Well, according to general ecological principles, species cannot coexist if they depend upon similar resources and seek those resources in the same place around the same times. And that raises the issue of how the numerous grazing ungulates that one observes in African savanna ecosystems are able to persist all on the same basic grass resource. The most generally accepted explanation is that niche separation is brought about firstly by body size differences and secondarily by relative mass dimensions. That means that smaller ungulates need higher quality diets for metabolic reasons, but they are also able to subsist on smaller amounts of food than the larger species. And also species with broader muzzles can crop short grass more efficiently than those with narrower muzzles. What that means is that the larger species are then dependent upon areas retaining relatively tall grass, while the smaller species benefit from the shorter grass that can be promoted by the grazing patterns of the larger species. And that's the general explanation that has been advanced for coexistence among grazers in East Africa, like East African regions like the Serengeti. But in this context, the smaller species, like gazelles, are not necessarily less abundant than the larger species like wildebeest and buffalo. In our case, we were dealing with three species of grazing ungulates that were fairly similar in size, and they also similarly seem to favor relatively tall grassland, which was quite widely available. There were differences among them, though, that could be important. For example, the zebra is a non-ruminant, and therefore should be more tolerant of fiber than ruminants are for digestive reasons. Buffalo move in very large herds, which is also going to affect their space use patterns and potential to select food.
And what was your hypothesis for the coexistence of the rare sable antelope and the zebra and the buffalo as the abundant grazers?
Yeah, the niche breadth hypothesis, as usually advanced, suggests the rarer species are specialists for a narrow range of resource types that are sparsely available and not fully exploited by more common species. So they tend to be the specialists. The interesting concept related to resource availability that we picked up actually from the plant literature is how rarer species might precisely exploit patches that retain favorable resources in between the depletion zones generated by the more widespread and more tolerant grazers in our case. And the quick question then for us is what form those resources might take. Could they be distinct grass species, different grassland structures, or something about habitat conditions? So our study was designed to cover a range of patch scales from feeding stations through to habitat types within the regional landscape.
Aha! And how did you conduct your study?
Well, we had earlier started a study focused on sable antelope to help explain why sable and other rarer antelope species were declining in abundance in Kruger National Park, and we've been trying to track the movements of sable herds using conventional VHF telemetry, where you have a directional antenna and you drive around giving a beacon signal, beep, 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 and try to find where that animal is. And then at some later stage, you try to find another animal. And that is quite tedious. And then GPS telemetry became available to us, initially on a trial basis, and then more collars became available. And we realized at that time that technology would enable us to compare what sable were doing with the buffalo and zebra herds that were thriving, despite all three species being dependent on fairly similar grass sources. So now we could simultaneously, with the GPS, simultaneously determine the locations of animals of all three species at the same time in the same region. And more than that, I should mention that we were using GPS collars that worked through the GSM or cell phone system. So the collars determined animal locations from the satellites through the GPS system. They then sent an SMS with that location data to a website. And in the field with a laptop, we could actually download where the animal had been an hour or two earlier, and then drive around, walk through the veldt with a handheld GPS, and end up at the exact place where the animal had been feeding, and in that way see exactly what grass species had been eaten. So that technology was crucial in enabling us to conduct a study covering 3 different animal species across the full range of patch scales, from what grass species have been eating somewhere in the middle of Kruger National Park to their space use patterns. Yeah, what I must say here is that the field research was undertaken by Dr. Valerio Macanza, and that was towards his doctoral thesis. He's now a lecturer in wildlife ecology at Eduardo Mondlane University in Mozambique, and my third co-author, who was a postdoctoral fellow at the time, contributed to the study around issues of habitat selection, a wildlife researcher in New Mexico.
So, the GPS technology sounds immensely useful for such detailed studies of habitat and resource use. How did the grazing habits of sable antelope then differ from zebra and buffalo?
Well, our main finding was that sable selected more strongly for patches of greener grass than zebra or buffalo, and also for the grass species that tended to remain greener, which is an unexpected thing. They were the smaller of the 3 species, and being ruminants probably required a higher quality grass component in their diet. But I should emphasize here that we were focusing our study in the dry season period when green grass would be limiting. In the wet season, of course, there would be plenty of green grass. We did find habitat partitioning between sable and zebra, with the zebra favoring more open savanna woodland on basaltic soils, which might actually be expected to be the more fertile substrate, while sable tended to occupy more wooded areas on sandier soils. And the buffalo herd more broadly utilized the whole study area, but they shifted towards the vicinity of the river during the course of the dry season because of their greater water dependency. There were pools that remained in the river during that period. The sable and zebra, on the other hand, carried on grazing mainly in the upland regions, and they traveled to and from water several kilometers. In the case of the sable, up to 6 or 7 kilometers, but not on a daily basis. The sable drank less frequently than the zebra, only every 3 or 4 days.
So, sounds like your findings could help explain how the sable coexists with zebra and buffalo. And how did your results fit into the general concepts of species coexistence theory?
Well, it seems to us that the sable were following the pattern described for plants, but of course as mobile herbivores, that they were able to precisely locate the places where some green grass still remained in the dry season despite the grazing pressure from more abundant and more tolerant species. But sable herds had been more widespread in the past. As I mentioned, we were studying them because the population had declined, and zebra and buffalo had been less abundant earlier in time, so we could not exclude the possibility that the sable had been displaced from using the habitat more broadly by increasing in the abundance of particularly zebra in the region. And that's related to a water point story that we've written about before. But the interesting thing is that it's not that it's either niche breadth or resource availability. Both concepts come into play. We can't say whether the sable are more specialized or more tolerant, in the sense that they need more green grass. They have a narrower niche. That just follows from physiology. But to be able to exploit tall grass, which is quite fibrous, they have to be tolerant, and they actually utilize a wider range of grass species than the more common zebra and buffalo did. So these concepts don't fit as neatly as theory might suggest.
And you mentioned that the sable antelope population in this area has declined. How is the situation of this rare antelope likely to develop in future?
Our concern now is that we've looked at reasons for the decline. We've looked at the habitat use. In the national parks, people have closed some of the water points to try to reduce the pressure on the sable, which has actually come partly through predation promoted by the increase in other prey species for lions. But you don't see any recovery. We have very little information on the herds now that we're not studying them, but the herd sizes seem to be remaining small, and they're not increasing despite more favorable rainfall and other changes. And we fear now that the herd sizes are so small that they are more vulnerable to predation. The females are less able to defend their young against predators, not only lions, but hyenas, maybe even wild dogs as well. So there's a real concern that the population is now at a low level when recovery might or might not happen.
Well, thank you very much for this enlightening interview. Let us hope for a brighter future for the sable antelope. You can read the full paper, entitled Habitat and Resource Partitioning Between Abundant and Relatively Rare Grazing Ungulates, in the July issue of the journal Zoology. We have come to the end of this episode. Thank you for listening. Please join us again for the next podcast to hear about more of the interesting research recently published in the Journal of Zoology.